This video tutorial is all about the structure of mammalian muscle. We're going to look at involuntary smooth muscle, the cardiac muscle, and then voluntary skeletal muscle. So first of all, the smooth muscle. Involuntary smooth muscle consists of individual cells that are tapered at both ends. So they end up looking spindle-shaped. At rest, each cell is about 500 micrometers long and 5 micrometers wide. Each cell contains a nucleus and bundles of actin and myosin. Now we're going to look more at actin and myosin in a later tutorial, but this image is just introducing you to those two protein types. Involuntary smooth muscle contracts slowly and regularly. It does not tire quickly, and it's controlled by the autonomic nervous system. Involuntary muscle is found in the walls of tubular structures, such as the digestive system and blood vessels. The muscle is usually arranged in longitudinal and circular layers that oppose one another. Cardiac muscle forms the muscular part of the heart. The individual cells form long fibers which branch to form cross bridges between the fibers. These cross bridges help to ensure that electrical stimulation spreads evenly over the walls of the chambers. When the muscle contracts, this arrangement also ensures that the contraction is squeezing rather than one-dimensional. The cells are joined by intercalated discs. These are specialist cell surface membranes fused to produce gap junctions that allow free diffusion of ions between the cells. Action potentials pass easily and quickly along and between cardiac muscle fibers. Cardiac muscle contracts and relaxes continuously throughout life. It can contract powerfully and does not fatigue easily. Some muscle fibers in the heart, such as the Purkinje fibers, are modified to carry electrical impulses. These coordinate the contraction of the chamber walls. The heart muscle is myogenic. It can initiate its own contraction. However, the rate of contraction is normally controlled by the SAM. Cardiac muscle appears striated, i.e. striped, when viewed under a microscope. Finally, the voluntary muscle. Voluntary muscle, or skeletal muscle, occurs at the joints in the skeleton. Contraction causes movement of the skeleton by bending or straightening the joint. The muscles are arranged in pairs called antagonistic pairs. When one contracts, the other elongates and relaxes. The muscle cells form fibers of about 100 micrometers in diameter. Each fiber is multinucleic, meaning it contains many nuclei, and is surrounded by a membrane called the sarcolemma. Muscle cell cytoplasm is known as sarcoplasm and is specialist to contain mitochondria and an extensive sarcoplasmic reticulum, which is a specialist endoplasmic reticulum. The contents of the fibers are arranged into a number of myofibrils, which are the contractile elements. The myofibrils are divided into a chain of subunits called sarcomeres. Sarcomeres contain the protein filaments actin and myosin, as we saw previously. Actin and myosin are arranged in a particular banding pattern, which gives the muscle a striped or striated appearance. Dark bands are known as the A bands, and lighter bands are known as the I bands. Voluntary muscle contracts quickly and powerfully. It also fatigues quickly. Skeletal muscle is under voluntary control. Its contractions are stimulated by the somatic nervous system. The junction between the nervous system and the muscle is called the neuromuscular junction, and it has many similarities to the synapse we've looked at before. So when an action potential arrives at the end of the axon, it causes calcium ion channels in the membrane to open. Calcium ions will then flood into the end of the axon. Vesicles of acetylcholine move towards and fuse with the end of the membrane. Acetylcholine molecules will then diffuse across the gap and fuse with the receptors in the sarcolemma. This will open sodium ion channels, which allow sodium ions to enter the muscle fiber, causing depolarization of the sarcolemma. A wave of depolarization spreads along the sarcolemma and down transverse tubules into the muscle fibers. Some motor neurons stimulate single muscle fibers. However, many muscle neurons divide and connect to several muscle fibers. All these muscle fibers contract together, providing a stronger contraction, and this is called a motor unit. The electrical activity of muscles can be investigated using an electromyograph. When a muscle is stimulated, the motor neuron creates action potentials in the muscle fibers. Electrodes applied to the surface of the skin detect the combined effects of these action potentials. A simple contraction of the muscle is seen as a series of apparently disorganized peaks on the trace. However, the amplitude of the EMG recording reflects the number and size of the motor units involved in the contraction. So, a more powerful contraction is seen as a higher amplitude. Placing electrical leads on the forearm, as shown on the slide in front of you, allows activity in the forearm muscle to be recorded. The recorded trace is called an electromyogram, and I've included an example of an electromyogram on the slide in front of you.